Let everyone join. We'll start within 2 minutes. And we'll cover the spermiation or release of spermatozoa. Just a moment, still people are joining. Okay, so I think we should begin now. Okay, so can we begin? So we have discussed this. So just for your understanding, this is where the diagram, if you want to, I'll also show you the separate PPT, but as of now, while we are understanding the textbook part and the question part, I'll explain it from this. So you can see over here, these are the Sertoli cells which you see over here. Okay, that is the Sertoli cell this way. Okay, then these are the primary spermatocytes. Okay, then these are secondary spermatocytes. Then you have this sort of structures which is spermatid. Okay, and finally this, as you see, this head is embedded in the Sertoli cell. Okay, if you see over here, the head is embedded in what we call, this is, let's say the yellow part is the sertoli, and this you see is the, it is embedded in that. Okay, and then finally the release, when the sperms are released, they are referred as spermatozoa. Okay, finally the spermatozoa will be released and they'll be embedded for obtaining nutrition into the sertoli cells. So spermatids will differentiate because of the growth factors and hormonal changes, as well as because of the nutrition provided by the sertoli cells. Okay, so this is sertoli cell, as you can see over here. Okay, this is the primary spermatocyte, as I am just labeling it out. Okay, then this is secondary spermatocyte. Okay, and then this is, as you see, this is spermatogonium. As you see, this is spermatogonium, this one. So any problems in this diagram? What I explained, guys? So you will be reading this, that's why. Now what we talked about is, over here, let's say this is spermatogonia, so it is 2n. As you can see, it is 2n46. Now there is mitosis, so you know mitosis, there will be two same cell. This is 46, this is also 46, okay, which are primary spermatocytes, so they are also 46. Okay, then there is first M1 or meiosis 1, so these are N. Okay, so meiosis produces reduction of the chromosomes. The first division is reduction, so the secondary spermatocytes will be 23, N equal to 23, as you can see. Then there is M2, which is like mitosis. Okay, so haploid, 4 haploid cells are produced. You can see over here 2 are there, over here we have 4. From this 4 there is differentiation, and then the sperms or spermatozoa are formed, the male gametes or germ cells, which will have 23, which are haploid. Gametes are haploid, as you know. Okay, as you know, gametes are haploid. So any problems in this, guys? Any problems in this? Anyone?
No, sir.
So this is the mature sperm image. Okay, so what I had just shown you how the shape they will assume, this shape. Okay, so this is where the, after the formation of sperm, spermiogenesis, sperm will be formed in this sort of structure, as you can see. Okay, so this sperm is released, okay, into the lumen of seminiferous tubules in this permeation, during this permeation. Okay, so I'll explain the structure, but just first we'll talk about the third phase, that is the spermiation. Okay, so the embedded sperms in the Sertoli cells. Okay, so the embedded sperms, as you can see over here, these are over here, these sperms are embedded in the Sertoli cells. Okay, you can just look at this part. The sperms is embedded in Sertoli cell. So this will be released in the, what we call as this permeation. So we're talking about the third phase, that is permeation. Okay, so what it says that the sperms or the spermatozoa, you can use both, means the same. Okay, so the are released luminar cavity of seminiferous tubules. Okay, And then they will go to where? Anyone? You remember from where? From this, they will be carried to which part? Anyone? Will be going to testis, then to the vasa efferentia, then to the epididymis, and then to vasa differentia. Okay, so that is the separate pathway. Okay, so after spermiation, this will happen. So they will be released from the Sertoli cells because their heads are embedded in that. So now they will be released from this condition, and then the sperm will be in this shape. Okay, so this is how they released from Sertoli cell. Okay, so this is how, as you see, the sperms are released in this condition. This is the image of a mature sperm. This is the image of a mature sperm. Now, over here, just try to concentrate on what I say. Just listen to. Okay, so see, basically, in this condition, this is the outer covering this is the plasma membrane. Okay, then the front part, this part, it is acrosome. Okay, over here you see nucleus. This part is entire nucleus. Okay, and we call this entire region, which has acrosome and nucleus. Okay, this entire part till this part is a head. Sperm has three parts. Then there is this part is neck. In this neck part, you will see something termed as centriole and mitochondria. Okay, let me highlight. If you can see this part, this part. Okay, these are mitochondria. That is mitochondria. If you can see this red dot which I have done, that is centriole. That is centriole. This part you see, that is centriole. This as you see, mitochondria are arranged in this spiral manner. Okay, they're arranged in this spiral manner, so they form the mitochondria spiral. This outer covering which you see outside the mitochondria, it is Nabenkan. Okay, it is Nabenkan. Okay, this is made up of flagella, what we call as the tail part. Okay, and this central filament, this line which you see, this black line which you see, that is termed as axial filament. That is termed as axial filament. Okay, so neck has only centriole middle piece, which is this one which we call as a middle. Oh, just a moment. Middle piece has mainly this part, mitochondrial spiral. Okay, so usually this is the sperm structure as given in your textbook. Okay, so now I will draw and then explain to you the same thing so you can have a better idea. So any problems in this, guys?
No, sir.
Okay. In exam you have to draw, so you should know how it is. So basically, This manner you can do it. It will not be as good as pen and paper, but then also let me try. Okay, then so this is the till this region. This region. It is the head part. Okay. Then this is the middle piece. This is the what we call as middle piece. And then this is the tail part. Got it? Over here, there will be two centriole. These are two centriole. I'm putting the dot over here. Okay. The acrosome, which is formed by Golgi body, is in this part. The acrosome will be in this part. Then there will be this region will have a nucleus. So this is plasma membrane. This green is acrosome. Okay. This is nucleus. Okay. This is acrosome, as you see, the nucleus. Then this part which you see, these are centriole. Over here, mitochondria will be arranged in spiral shape arrangement in this manner. Okay. These are all what we call as mitochondria arranged in this spiral position. As you can see, okay, they have two-layered structure, as you know. So this is what we call as the mitochondria. Just so many mitochondria will be arranged over there in this region. The covering is Nebenkan. Then we have this is flagella. Okay, so this is how you can differentiate different parts. So that's why I've shown the head, the middle piece separately, and the tail. It can move by flagella. Energy is provided by mitochondria. It needs many mitochondria. As you can also see over here, in this, if you count the mitochondria over here, look at this part, then 10 odd mitochondria are there. Okay. So it needs move energy for fertilization. So these sperms can move. That's why they need this energy, and it comes from ATP through mitochondria. So it's the energy source of swimming. Also a question. Okay, so this is how you can draw. So any problems in this, guys, in this structure? Yes or no?
No, sir.
Okay. Moving on, the last part is about hormonal control. And then tomorrow we'll take questions from this. So I hope please come prepared. I will ask questions so I know where do you stand from this part which we have covered. So the last part is about the hormonal control of spermatogenesis. Okay, it is about the hormonal control of spermatogenesis. Okay. So you must have learned endocrine system in 11th. Okay, so I'm assuming that you are aware about that. So there is hypothalamus. Okay, it is in, as you know, it is in contact with the pituitary gland. Okay, which part of pituitary? Especially anterior part. Okay, I hope you know about nervosa and distalis, pars nervosa and distalis of pituitary. So it releases what? GnRH, which is, anyone can tell me the full form of this? Anyone? I hope you had learned this in 11th. It is gonadotropin releasing hormone. Okay, so it works, it basically acts on the pituitary gland. And now this pituitary will show its effect on Leydig cells and Sertoli cells. Okay, so the secretions of pituitary are mainly through FSH and LH. Okay, so there will be secretion of FSH and LH. Okay, so wait, so what will happen now? I'll just show you the second part of this. So what will happen now about this? So as I had talked to you that there is hypothalamus. Okay, from then it is affecting the anterior lobe of pituitary, and then pituitary is releasing two different hormones. That is, one is LH and FSH. Hypothalamus over here, GnRH comes up as we have talked over here. Okay, so this will affect Leydig cells, and this will affect Sertoli cells. LH means luteinizing hormone. Okay, FSH means follicular stimulating hormone. So under the influence of LH, these Leydig cells will secrete testosterone or male sex hormone, or what we call as androgens. Okay, and this will affect reproductive tract, especially in male. Okay, now the Sertoli cells, it is FSH is affecting Sertoli cells. So Sertoli cells can secrete two things. It can secrete two things. That is, one is it can secrete certain factors. It also secretes a hormone which is inhibin. Okay, so it can secrete Sertoli cells as these two roles. Now, what will happen? Now, this testosterone can also, what it can do, these factors, okay, basically can lead to, can stimulate spermatogenesis. This can stimulate spermatogenesis, as you can see over here. This testosterone will also affect and will increase spermatogenesis. Okay. Got it? So in this manner, this is some growth factors as this is referred, okay? From Sertoli will also play a role in stimulating spermatogenesis along with the testosterone effect of the Leydig cells. Now, what will happen? Now, this is the general process. Okay. So androgens will basically stimulate spermatogenesis. FSH will lead to production of some factors which helps in this. Now there is one question about this inhibin. There is one question about this inhibin. When there is excess FSH, when this FSH is produced more than required, okay, when this is produced in more than required, okay, when it is excess, then what will happen? In excess FSH, what will happen? This inhibin, okay, will go on and act onto this anterior lobe of pituitary. Will go on and will act onto this anterior lobe of pituitary. Will stop synthesis of FSH, and if FSH synthesis is stopped, then ultimately the testosterone and the spermatogenesis process will also stop. So this is termed as a negative feedback. Inhibin is responsible for negative feedback control. If you learn the feedback control in the endocrine system. So if anterior pituitary will stop secreting FSH, then directly GnRH will be stopped. Okay, so when there is excess FSH GnRH, inhibin will be produced and it will stop and it will regulate this spermatogenesis. It will stop and regulate this spermatogenesis. So this is just what it is once again. So what it is trying to explain, that mainly hypothalamus okay, let me take the pointer. Hypothalamus will secrete GnRH, will act on pituitary anterior lobe, so it will secrete FSH and LH, that you know. So that is what I have mentioned over here. Now, whenever there is normal, when the spermatogenesis is required, in that condition, LH will be acting on Leydig cells. Okay, then FSH will be acting on Sertoli cells, as you can see over here. Okay, GNI is produced by hypothalamus. Okay, this will all act on anterior pituitary. Now, these Leydig cells can secrete testosterone, and it will affect the testes and reproductive organs. So this will stimulate the spermatogenesis, as you can see. But when and these Sertoli cells can secrete these growth factors as well as inhibin. But when there is excess of FSH, in that condition, this can have a feedback control. This can go on and have a feedback control on anterior pituitary. It will affect the anterior pituitary. So whenever there's excess of FSH and LH, and whenever this process is to be stopped, this inhibin can bind to the anterior lobe. Okay, it will basically suppress the FSH release by acting onto the anterior pituitary. So this is how the negative feedback control is inserted, and the entire process of testosterone production, or in short, you can also call it as a androgen production. Okay, because this secretes mainly androgens. Okay, the entire process of androgen production, or the common androgen is testosterone. It will be controlled, it will be regulated in this manner by inhibin through negative feedback. So this is all we have in male reproductive system and gametogenesis. Okay, if you have any problems, you are free to ask. If you have any problems, you are free to ask. 3 phases: spermatid formation, spermiogenesis, permeation. Spermatid again has 3 phases: multiplication, growth, maturation. Okay, meiosis and mitosis will lead to formation of the spermatids. Then we have polarized spermatozoa, the changes which will occur, Nebenken and all those things we discussed. Okay, we talked about this entire process and the sperm structure. So now if you have any problems, you are free to ask. When the sperms are released, they are in this condition. This is the what we call as the mature sperm. Okay, so if you have any questions, you are free to ask. Otherwise, this is all for today. Tomorrow we'll meet at 8:30. We'll take questions. I'll ask you questions, so come prepared. I should know where do you stand and how what you are doing. Okay, so how you, and we'll solve the previously asked questions and all, so you should know. Also other questions I will ask, and then we'll go on further. So if you have any problems, please feel free to ask. Anyone, any issues, guys?
The function of GnRH hormone.
You're not audible better clearly. I heard GnRH, it means gonadotropin releasing hormone. It is released from pituitary gland. I hope you learned endocrine system in 11th. Okay, it regulates the secretion of pituitary gland. It is in contact with the anterior part of the pituitary gland. So if this will act on pituitary, then only this will release FSH and LH. Okay. So as you know, pituitary is controlled by releasing hormones from hypothalamus. Okay. As you know, pituitary is in control of hypothalamus, which releases neurohormones, which can be inhibiting or releasing. This is releasing, so it will stimulate pituitary to produce FSH and LH. So it will stimulate pituitary to produce FSH and LH. Okay, go through endocrine system once in 11th. Okay. So anything else? Any other questions? Anyone? I will share the notes in the group. We'll also upload lectures today or tomorrow. So go through that. So this is all for today. Thank you, guys. We'll meet tomorrow at 8:30 a.m. Please, everyone, be on time.